I'm going to go ahead and get started, Mark. And so thank you, everybody, for joining us for our Department of Internal Medicine Thursday Roy Benke Grand Rounds. Really, it's a pleasure to have Dr. Mark Glaum, uh, who's Associate Professor of Medicine and Pediatrics in the Division of Allergy and Immunology. Here for 21 years, hard to believe. I was looking at Mark's CV going, how on earth has it been 21 years? It just seems like it was yesterday when I met this very young allergy professor, assistant professor at that time, who came from Hahnemann, where he had received his MD and his PhD. Um, Mark, as he mentioned, went to Fordham for his university work. But Mark arrived in 2004, joined Dick Lockheed's incredible allergy immunology division with Roger Fox, Dennis Ledford, and so many others. And Mark was really the young kid in the block. So it was just really uh, a recharging, I know, for the division. Mark has done nothing but been an absolute incredible scholarly activity worker, such an important part of the annual allergy and immunology presentations. Mark has done many other national presentations and publications, served as program director of our incredible allergy and immunology fellowship for nearly a decade, and it remains a incredible vital part of our faculty. So also his wife taught my kids at the Academy of the Holy Names, so I've gotten to know the entire Glaum family. So it is really a pleasure to introduce Mark, who again, hard to believe, has been here for a generation, and he's talking today about such an important topic, one that my house staff were noticing me the last 2 weeks or so on rounds, sneezing and saying I have to go to the bathroom and blow my nose. Uh, it's been an horrific allergy season, so this timing of this talk is great. And Mark's talk is just, his title is great. Florida Allergens. Why is my patient sneezing so much? Or as my residents say, why is my attending sneezing so much? So Mark, take it away, please.
Well, thanks, Dr. Lozama, for your kind intro. Yeah, it is hard to believe it's been 21 years now. Not so much the young kid on the block anymore. But this is a very important topic for allergists, but also I think for other physicians and specialists to appreciate. There are some unique aspects to allergy and immunology, the specialty I love. One of the unique aspects is that you can enter the fellowship from either internal medicine or from pediatrics. So as such, in our fellowship training program, you're prepared to see both kids and adults when you come out. And in fact, our practice is probably 20 to 30% children. So I see patients from infancy up through their 80s or 90s. And so it gives quite a breadth of exposure and keeps things, fresh and new, seeing different populations of patients every day. The second, another unique aspect of allergy and immunology is that we're one of the few specialties that really helps patients navigate and manage their symptoms that are in response to the environment. And so if you think about it, what allergy really is in a predisposed individual is that their immune system gets tricked or hijacked to start to recognize harmless items in the environment, like pollens or dust or mold or animal danders, as potential invaders. And so allergy occurs because the immune system mounts a response to help fight off these invaders and utilizes mechanisms in the immune system, usually geared towards parasite infections. But those inflammatory responses cause the runny nose, the sneezing, the congestion, the itchy, watery eyes, the wheezing in the case of asthma. And so if you're going to help your patients navigate their battle with these environmental allergens, you really have to know something about the environment. And every allergist, when they go into practice, really needs to take time and appreciate what are the important allergens and seasons in the area where you're practicing. And in fact, when Dr. Lockey came to Florida to help found the Division of Allergy and Immunology, really very little was known about what were important allergens in the Tampa Bay area. And much of the early work in the division went to characterize and identify the important and the not so important allergens in the area so that we could help patients with these symptoms. So at the end of the talk, if anyone's interested, I have probably 10 or 15 pages of references from over the years, really just having to do with allergen identification that have come out of the division. So we'll get started. So we're going to talk about common Florida aeroallergens that are responsible for allergy symptoms. We're going to talk about the physical characteristics of different pollens and how that helps you identify them when you do pollen counts. We'll describe how air samples are collected and analyzed and how that's different than what you see on the news insofar as pollen forecasts. So we'll go into a little bit about that. What we actually do is count the actual pollen grains in the air samples. And then we'll talk at the end about some cross-reactivities among pollens and also between pollens and foods that causes an interesting problem where patients think it's food allergy, but it's really more of a cross-reactivity issue. So we'll go through an introduction. We'll talk briefly about how you sample the air and examine the pollen counts. We'll talk about important pollens and seasons as it relates to tree pollen, grass pollen, weed pollen, as well as molds and fungi. And then briefly go over allergen cross-reactivity. So aeroallergens are really airborne particles, and they have certain features that make them more or less allergenic. Buoyancy is one of the important features of pollens that cause allergies. And it really has to do with the different, depending on the plant, they are pollinated by different methods. So plants can either be insect pollinated, wind pollinated, or a combination of both. And from an allergy standpoint, the ones that cause most of the problems in allergy are really pollinated by the wind. So the history of your aunt who walks into a hospital room where there's flowers that someone's delivered, and they start sneezing. They think they're allergic to the lilies or whatever the plants are. That's really not the case. It may be more just the odor or the fragrance causes an irritant reaction, because the pollens made by those ornate flowers really don't cause allergy. And not all plants cause pollen or create pollens that cause allergy issues. For example, pine trees, although pine pollen is a majority of what you see covering your car in green and yellow in February and March, the pine pollen is really not that all allergenic because it's very large and heavy, and most of it falls to the ground. So general characteristics of pollen. So pollen is the male gametophyte, single cell. It's surrounded by an inner and an outer wall and aids in plant reproduction. Particularly the airborne pollens are designed to be buoyant and to remain aloft in the air. That makes it more difficult to capture on a surface. So airborne allergenic pollens are typically small, so they're in the range of 20 to 60 microns. And that size allows it to very efficiently deposit in the conjunctival or in the nasal epithelium or in the upper airways. They're generally too large to make it all the way out to terminal airways, at least in whole fragments, or at least in whole pollen grains, but fragments could potentially get deeper into the lungs. And fragmented pollen that occurs after thunderstorms, for instance, can release starch granules and make it into those lower airways. So the main types of dispersion, we talked about either air vectors or insect vectors. Anemophilus refers to airborne pollen, and plants that make airborne or that are pollinated by wind, they tend to have an absence of bright colors, scents, or nectars. They have kind of ugly grouped flowers together, mostly anthers. The pollen grains tend to be small, as I mentioned, 12 to around 50 microns. They have very little surface lipid, but they do have unique shapes to them. They're not all just circular little grains. And because they rely on the wind to pollinate them, they really overproduce pollen because there's a lot of wastage. But the pollen grain does travel far. So for instance, one birch tree in a pollination season makes about 1 billion with a B pollen grains, but the dominant tree in our part of Florida, live oak, makes 5 times that. And we'll talk about why our season in Florida is so long. And that compares to entomophilous or insect pollinated plants, which are really ornate flowers. They're large, colorful, with scented nectars to attract insects. They don't make very much pollen because they don't need to waste it. So it's very sticky, so that the insects come in for the nectar. The pollen sticks to the insect, so the insect can transfer it to the next plant. They make relatively little amounts of pollen that's heavy and sticky, as I mentioned. And then there's amphimophilous, which are plants that are both rely on airborne and insect vectors. So in thinking about allergenic pollens, you're thinking about wind pollinated plants. A lot of the pollen output from these wind pollinated plants is lost by fallout, but because the particles are so buoyant and light, they can really be carried hundreds of miles. So ragweed pollen in the season has been found hundreds of miles out to sea. And there have also been reports that depending on the jet stream wind currents in February, when Texas cedar pollen is really hitting its peak, it can be detected all the way up in Canada when nothing is growing. So it's not so much what plants are on your street or around your house, although that does make some difference. It's really a regional effect because the pollen gets carried on the wind for miles and miles. So this kind of outlines our allergy seasons in Florida. And really we have, compared to other parts of the country, we have one main season, and that's the tree pollen season. You can see on the top bar, the darker gray, the season begins some years in late November or December. There's a minor seasons in that lighter color, white. We see elm and some other trees come out maybe October, November. But really the season gets going in December, January. It typically builds to a peak in March, and depending on the year, it can drift all the way into late May or June. Compared to other parts of the country that have a big grass pollen season in the summer and then a big ragweed season in August through the first frost, we do have some grass pollen and weed pollen around, but it pales in comparison to the amount of pollen that's produced by trees. So again, our major tree pollen season, December through May, primarily driven by oak pollen, but you'll also see pine, Australian pine, cypress, and cedar. That's probably number 2 importance behind oak, as well as bayberry and mulberry. There's a minor tree season in the fall with elm now. Used to be Australian pine, but there's not as much of that around anymore. Then we have moderate levels, mild to moderate levels of grass pollen, which is primarily April through October. Weed season, mainly driven by ragweed, the summer through into the end of the year. And then molds are present all year round. They tend to be higher levels during the rainy season, but we never get a hard frost to kill off the mold, so they're present any time of year. So how do we detect what's in the air? There's basically 6 methods of aeroallergen sampling. Sedimentation or gravity sampling, not very efficient. Rotating arm impactor sampling, impinger sampling, centrifugal or cyclone sampling, filtration sampling, and then the most common, what's used most commonly is a suction or cascade impactor sampling. So volumetric samplers are really the standard of what's used. And this really is a device that needs to be at a high elevation above the second story. Between 2nd and 5 stories is thought to be optimal. And it really makes use of a vacuum air pump that sucks air into a device that's directed into the wind. And it has an advantage for detecting smaller particles like molds and the smaller pollens. And the key is that you can quantify the pollen counts as pollen grains per cubic meter of air. So it really gives you a standardized quantification advantage. Some examples are the Hirsch spore trap, which is good for measuring diurnal variation. Burkhardt spore trap, which is what we used at the pollen sampling station at USF. And this is really an adhesive tape that goes around a drum and can be measured continuous sampling for a whole week, or depending on the head that you put in the machine, we typically do it over a 24-hour period where the pollen or air sample gets impacted onto a grease-coated microscope slide. And there's Anderson samplers where air passes through sieve-like plates with decreasing diameter holes with petri dishes at each level. So this is the Burkhardt sampler. This is the standard sampler that's used throughout the country and advocated by the National Allergy Bureau. It's a smaller, lighter version of the Hirsch sampler. And as you can see, I don't know if you can see my pointer, but it has a fin on the back that directs the front of the device into the wind. At the bottom, near those 3 feet, is a air pump that sucks air through this little orifice in the front, and then impacts the air sample onto a greased microscope slide over 24 hours, or this coated plastic tape over 1 week. It's great for small particles and best for outdoor sampling. So where can you look at and see where pollen counts are throughout the country? So if you go to our main academic organization for allergy, which is 4 A's and an I, AAAAI, and their website is at the bottom, www.aaai.org. And you look at the top of the homepage, and you'll see check pollen counts. And if you click on check pollen counts, you can see the different NAB counting stations throughout the country. So here you can see how the pollen grains are counted as pollen grains per cubic meter of air. And if you click on the Tampa station from earlier in April, and then look down under trees, weeds, grasses, and molds, you can see this particular week, tree pollen was high concentration at 233 pollen grains per cubic meter. Now, we typically start to see patients come in with symptoms if they're tree pollen allergic when the counts get to about 50 to 100 pollen grains per cubic meter. And during the height of the season in early March, most years, the count will be in the thousands. Some years 1,000, some years up to 3,000 or 4,000 pollen grains per cubic meter. And those are years when we get inundated with people coming in with itchy, watery eyes, sneezing, congestion, and really quite miserable, even though they're on a ton of allergy medications. If you compare that to the weeds and grasses, you can see the counts are much lower, although this isn't the peak season for them. But the counts for weeds and grasses typically don't go above 20 or 30 pollen grains per cubic meter. So that's why we don't have as big of a grass pollen season or weed pollen season. There's just not as much of grass and weeds around that make allergenic pollens. Compare that, though, to mold. You can see the mold count that week was over 40,000 mold spores per cubic meter. So the levels that we must have gotten rain that week, because usually it's around 7,000 or 8,000. But if we get rain, then it can go up into the tens of thousands or even hundreds of thousands. But interestingly, during the rainy months in Tampa, when the counts are up in the tens or hundreds of thousands most days, we don't get inundated with people coming in with nasal allergy symptoms. So that tells us that at least molds in the form they are in Florida don't seem to be as big of an allergy issue as the pollens and dust mites and animal danders tend to be. And compare this to perhaps what you see on the Weather Channel or on pollen.com or some of the other news channels where they give pollen forecasts that go up to, they predict, it's going to be a 9 or a 10 later in the week. Those are really not based on pollen counts that year. So many times they're off and incorrect. And the reason is that they go by historical pollen counts from past years and then combine that with the current weather forecast. So if it's going to be rainy for several days, that usually washes the pollen away. So the forecast will be lower. If it's going to be very windy and dry, the wind kind of picks up the pollen from the from the sidewalk and from your cars, and then redistributes it so the pollen forecast is much higher. But that's very different than actual getting pollen counts. So when we do a count, which we typically do once a week, the pollen air sample may be collected from Tuesday morning to Wednesday morning. Then it gets stained and counted and may get reported on Thursday or Friday. So the pollen count you see at the end of the week really reflects what was happening earlier in the week, just to keep in mind. And then if you scroll down further on the website, the National Allergy Bureau is the organization that kind of coordinates pollen counting throughout the country. And they have certified stations where individuals have to go through training and pass a test on accurate pollen counting. They have to have the Burkhardt sampler and upload their counts to the NAB site every week or several times per week. But you can see in the U.S., pollen counting stations stretch from Anchorage, Alaska, all the way down to Puerto Rico. And you can see there's probably at this point in time about 60 some counting stations throughout the country. And if you look at Florida, you can see us over Tampa, and we're really one of just 2 pollen counting stations in the state of Florida. The other one currently is up in Jacksonville. So, you're really only getting pollen counts in certain areas where these stations are set up. And here's our pollen counting station on the roof of the MDC building. It used to be on the roof of the research building at the VA until they started their renovation, which I think is going on 10 years now. But you can see several of the Burkhardt counters here on the roof. And then if you take the top off and the head out, this is the on the right side, you can see this kind of circular portion with a nub, and that gets wound up like a grandfather clock and actually ticks. And what it does is as it's wound up, you can see this microscope slide at the bottom here. That gets advanced 2 millimeters per hour and travels up to the top of the head. And it's in front of a horizontal slit on the device that impacts or sucks the air sample in, so it impacts on a silicone grease coated microscope slide. So you get a survey over a 24-hour period of what's been in the air. Then that microscope slide gets stained. So you can, the pollen is otherwise clear on the microscope slide, so it's stained pink, so you can identify the structures and try to identify which is oak pollen versus pine pollen versus cedar or cypress. And again, we do this currently about once per week. So sampling and examination, this really is done in most counting stations by direct microscopic examination, and that's how we've been doing it. I kind of reopened the pollen counting station about 10 years ago. It had been kind of dormant for several years prior to that. But we either me or another faculty member who used to be here in Tampa will count the slide once a week and upload the data to the NAB site. It's really the quickest way for detecting and counting pollen and fungal spores. There's other methodologies that we've looked at. We tried using PCR several years ago, got a small grant to look at that, but it was difficult because the other contaminants that are in the air samples kind of inhibited the PCR enzymes. Others have looked at chromatography methods for identifying pollen and other automated techniques. There are some out there, they're just very expensive, at least at this point, but maybe in the future they'll become more effective and we can have automated pollen counting without all the labor that's involved in collecting and counting the slides. Magnification under the microscope is 400 for pollen and 1000x for mold spores, which you need oil immersion. And the pollens are separated and identified based on their size, on their shape, on germinal apertures, their sculpture, and also the cell contents. So this is some examples of what the pollens look like and how they can be different. They can have no apertures and be smooth on the outside, like cypress here on the left at the bottom, or they can have these grooves that are a single, like a monocolpate, or they could have 3 of them as tricolpate. And each of these are a little bit different and unique depending on the pollen species. They can also have pores. So this example on the bottom right screen is grass pollen with a single pore, or tree pollen in the middle can have 3 pores, triporate. They can have axial pores that just run around the center, stephanoporate, or multiple pores, periporate. So we'll start with the main players for our seasonal allergy issues and what's making Dr. Lozama sneeze and others with seasonal allergy sneeze is mostly oak, because this is the dominant tree in this part of Florida. And in fact, if you're in the city of Tampa, you have to get a permit to cut one down, and they don't give the permits out very willingly. It's from the Fagaceae family and includes Quercus, which is oak, and beech. American beech is present from Nova Scotia to Florida Panhandle and out west to Missouri and Texas. The oak is in all lower 48 states except Idaho. Black and white oak are most important across the U.S. And it's not the acorns or the leaves or the bark that causes the allergies, but the pollen. And why do we have such a long season here? So in the Northeast, the tree season is kind of confined to maybe a few weeks to a month. And why is our season 6 months or 5 or 6 months? That's because there's more than one species of oak in the Tampa Bay area. There's actually 11 different species of oak, and they all tend to pollinate sequentially. So you'll have some that are early pollinators in December and January. Then the bulk of them will make their pollen sometime in March. And then you have late bloomers that kind of stretch out into April and May. And so that contributes to our 5 or 6 month tree pollen season. The other trees we'll talk about are also kind of mixed in, but in the same time period. But if you look at during the peak of the season on one of these stained pollen styles, it's just loaded with oak, and it's difficult to really count much else. So stained under the microscope, this is what the oak pollen looks like. It's got 3 pores and kind of has this warty appearance. The picture up top is more typical of what you see in a freshly stained air sample slide. Beech looks very similar because they're in the same family. It just has longer furrows. So pine is not as important from an allergy standpoint, probably because it's a very large size. Remember, most of the allergenic pollens are somewhere in the range of 20 to 50 microns, and pine is much bigger, so it's easier to pick out on a slide. Because it's so heavy, that most of the pine pollen falls to the ground near the tree base, but it's present from the Arctic all the way to the southern hemisphere. And pollen identification questions do still come up on in-service exams in allergy and on your board exams. And so for the fellows, the pine pollen has these 2 air bladders, and it's quite large, but it kind of reminds you of a Mickey Mouse hat, so it's easy to recommend or to recognize that way. There's no visual openings, but it has these 2 large air bladders. Cedar and cypress is probably the second most important allergenic pollen, and you really can't differentiate the cedar from cypress. They look the same under the microscope. But they represent important allergenic trees all across the U.S. The pollen grains are small, like most allergenic pollens, 25 to 35 micrograms. I'm sorry, microns. Mountain cedar is important in central Texas. In Tampa, we have red cedar, bald cypress, and pond cypress. And if you look under the microscope, it had the cypress, and cedar has this kind of smooth appearance, no pores and no furrows. It's got this kind of star-like center. And when you stain it, sometimes on the slide below, you'll see that the pollen grain ruptures, and you get this kind of clear center that kind of extrudes, and it looks like a Pac-Man if that happens, but very characteristic and easy to identify as well. Other important ones are bayberry. This is in the Myrica family. It's also called wax myrtle. It's on the coastal Gulf of the East Coast, Gulf states, and California. We have southern bayberry that blooms in February, March, and April. Historically, the leaves are very aromatic when they're crushed, so it was used in colonial times to make candles. And it has a unique appearance. It's kind of circular from one side, but triangular from the other side, and it's got 3 irregular germinal pores. Mulberry is another one. This is in the Moraceae family. Paper mulberry is more common. Red mulberry is what we have here. It's, again, a small pollen, 11 to 20 microns in size, and kind of has this barbell shape to it. Usually 2 to 4 pores with a thickened outer coat below the pores. Then there's minor trees. These really make less than 10 pollen grains per cubic meter on most weeks. Again, remember, oak is into the hundreds and the thousands during the height of the tree pollen season, but these are much less common. Australian pine used to be seen on the pollen counts when I first came to Tampa, but it was classified as an invasive species. So, the county goes around and cuts them down when they see them. It was originally brought in as a windbreak on farms, but it does make a pollen that people can be very allergic to. It may be more prevalent south of the Tampa area. But it has an elliptical shape to the pollen, about 24 to 33 microns, 3 pores, and a very thin outer coating. Elm is present in Mississippi and Ohio River valleys. There is American elm, and Chinese elm is used more as a decorative landscape, and that's probably more of what we see here. But this contributes to an early minor season, like October, November, but it is also seen during the peak season as well. And elm has this kind of 5-pore appearance, or tetraporate. It's kind of got a gap between the outer core and the inner core, so it makes it easier to identify under the microscope. Olive and ash, they cross-react. We don't really have olive here. That's more out west in California and Arizona. But others in the family, and along with ash, are privet and the olive. The privet becomes airborne when the tree is shaken by animals. In Tampa, we have water ash, and ash is present throughout the eastern US as well. And it has this kind of quadrangular tetraporate with short furrows, 20 to 30 microns in size. And olive and ash look the same under the microscope, and they cross-react allergenically. Then maple and box elder, these are in the Raceae family. Box elder is the most important allergenic maple plant in the US. Pollen size, again, in that allergenic range of 20 to 30 microns in size. And for identification purposes, this also comes up on exam sometimes. So it has this kind of beach ball look to it, or the trifoil radiation sign is another way to remember maple under the microscope. Willow is mainly insect pollinated, but we do see willow pollen grains on our counts during the season. Not as important as an allergenic pollen, but it is there, so it may contribute. Then we'll move on to grasses. Grasses are in the Poaceae family. There's over 600 genera and 10,000 species of grasses, but more than 95% of the allergenic grass pollens are in these 3 families. One is with fescue, rye, and timothy. The second is with Bermuda, and then the last is with Bahia and Johnson. The grass pollens all look the same under the microscope, so it's really difficult to differentiate among them, but they all have this single pore. They're a little bit larger, 20 to 60 microns, but they all have a similar look to them. Again, this is testable on in-service and board exams. And if you're a Star Wars fan, you can think of the pollen grain with that single pore looking like the Death Star from the initial Star Wars series. So when grasses go to seed, like Bahia, which is used in residential applications and medians of highways and whatnot, tends to have this large V-shaped spikelet when it goes to seed. That's when the pollen's being made. And this is Bermuda grass when it grows up, more common on golf courses and whatnot. But this is when it grows wild, that's what it looks like. And then Timothy grass, not so present in Florida. It's more of a northern grass. But Johnson grass is present in Florida, more so in the northern part of the state, but it does cross-react with Bahia. And you can see, depending on the family, there's cross-reactivity among those grasses. So the ones on the left, the Poetes, are the northern grasses, so they all cross-react. Johnson and Bahia cross-react from an allergenic standpoint, and then Bermuda cross-reacts with saltgrass and grama grass. The cross-reactivity can be exploited either during skin testing or immunotherapy. So if you don't have enough room in your immunotherapy mix to include all the grasses, for instance, you know that if you put in Bahia and they're sensitive to Johnson, that you'll get some cross-reactivity and likely some benefit because of that cross-reactivity issue. So we'll move on to weed pollens. So again, weeds not as important as the tree pollens in Florida, but they are here, just not as prevalent. Ragweed is the main one, and that's the Asteraceae family. Mugwort's also in the same family with ragweed. Then you have the chenopod's, pigweed, Urticeae, wall pellitory, Brassicaceae, and rumex, which are the sorrels, dock and sheep sorrel. So the ragweed family, Asteraceae, has over 13,000 species. They're throughout most of the world. The most important is ragweed. We just have short ragweed here in Florida. Other parts of the country have giant ragweed and other forms, so the levels tend to be much higher. In other parts of the country, ragweed season, like in the Northeast and the Midwest, seems to magically occur right in the middle of August, and then continues through the first frost. And that's really what coined the first term hay fever, because it was thought that it had to do with harvesting the hay on farms, because that's when they do it. But it also really is due to the ragweed that blooms and pollinates during that time of year. Others in the family include mugwort, dog fennel, marsh elder, and cocklebur. So here are some examples of short ragweed growing on the top panel. Another testable pollen identification is ragweed because it's very unique. It has these spikes or spines throughout, so it's very unique appearing under the microscope. Again, short ragweed is the only species in Tampa, but ragweed is responsible for most of the pollen in August and through November. It's tricolporate with these pores and furrows and the small spines throughout. This is what wild pigweed looks like. Pigweed and other chenopons include lamb's quarter, sagebrush, Russian thistle, burning bush. And the pigweeds and chenopods have another unique appearance. They tend to have these dimples that kind of reminds you of a golf ball. They're about 20 to 30 microns in size, again, right in that allergenic window. And most overlap with each other from an allergen cross-reactivity standpoint. Here's some examples of lamb's quarter and Russian thistle you may see growing in open fields. Sheep and dock sorrel in the rumex family, they sometimes have 3 or 4 pores, long, thin furrows. They have a thin outer coating and usually start blooming in March and go through into the summer. Here's examples of wild growing dock sorrel and sheep sorrel that you might see. And then to molds. So molds are present throughout the world except for the polar caps. They present in different forms. And so the allergenic form is really due to the spores that are ejected from their reproductive structures. And they really, as you remember, think back to the counts that we saw on the NAB site from earlier in April, the mold count was 40-some thousand mold spores per cubic meter. So they really formed the majority of suspended biologic particles in the air. But mold, although maybe in Florida not as prominent of an allergenic contribution, it does seem to affect human disease. So alternaria in particular, when levels are high, is associated with severe asthma. There's 5 phyla that contain the allergenic molds. Zygomycota, Mucor, Rhizoporus, and Aspidia. Ascomycota, which is Cladosporium, Penicillium, Aspergillus. Those are more the Penicillium, Aspergillus are more common indoors if there's water damage. That they constitute 50% of all fungal species. Then there's Basidiomycota, Deuteromycetes, and Oomycota, which are mildews. Cladosporium is the most prevalent outdoor airborne fungus. That's what we see at highest counts when we do the samples. It's widespread in temperate North America, and they can reach very, very high concentrations in the air. Aspergillus can play a major role in allergic rhinitis and asthma, but also in ABPA, as well as allergic fungal sinusitis. And as I mentioned, Aspergillus and Penicillium are more likely to be found indoors. They grow really well on drywall if it gets wet, or any other water intrusion into the home. You're likely to see Aspergillus and Penicillium. Penicillium, again, typically found indoors, cross-reacts from an allergenic standpoint with Aspergillus. And that alternaria gets to levels about 500 to 1,000 spores per cubic meter of air present in temperate grain-growing regions. Then Helmintsporium, also found in warmer regions, especially in the southern US. Aspergillus is a prominent cause of allergic fungal sinusitis, along with Helmintsporium. So moving on to cross-reactivity issues. So there can be cross-reactivity among pollens if they're in the same family. So again, you can take advantage of this if you're putting someone on an immunotherapy program and there may not be enough room to include everything they're sensitive to. You know that, for instance, if you include cedar, then that should cover their cypress and juniper sensitivity because they all cross-react. Remember, birch and alder and hazelnut cross-react. Olive and ash cross-react. Beech and oak are in the same family. Pecan and hickory, and then poplar, aspen, and cottonwood. All cross-reactivities. The grasses cross-react depending on which family they're in. And this is taken advantage of in certain preparations, such as sublingual immunotherapy tablets that are approved. So Aeroallergens and Grass Tech are the ones that are improved in the US. However, they never really made much penetration into Florida because we don't have these grasses in Florida. These are all northern grasses by and large. So, the only time we typically would use these for immunotherapy is if a patient is spending their summers up north and they have problems in the grass season up there. They may go on to the sublingual tablets for when they're traveling back home, and then can stop them when they come back to Florida. Bermuda tends to cross-react with western prairie grasses, and as we said earlier, Bahia and Johnson cross-react as well. Among the weeds, all the ragweeds cross-react, and there is a sublingual tablet for short ragweed called Ragwitech. Again, not as popular in Florida because we don't have as much ragweed here, but we do occasionally have patients on that sublingual tablet. Sages and mugwort cross-react, thistle, burning bush, lamb's quarter, all cross-react, pigweed and western water hemp cross-react. And there's another kind of interesting situation where pollens and foods can cross-react. And so this is a situation where you might see a kid come in for evaluation for food allergy because when they eat either certain fresh fruits or vegetables or nuts, they get this itching in the mouth and the throat, or tingling or itching on the lips, but it doesn't progress on to vomiting or generalized hives or breathing problems. So it's not a true IgE-mediated food allergy. And many times if you skin test them to these foods, they'll be negative. And what really happens is that if someone's very pollen allergic, depending on the pollen, proteins in certain foods look to the immune system like the pollen. And certain foods go with certain pollens go with certain foods. So for instance, in patients who are highly ragweed sensitive, certain fruits such as fresh melon, fresh zucchini, banana, cucumber, sunflower seeds, watermelon may cause this itchiness or tingling in the lips, itchiness in the mouth and the throat, but no vomiting, no generalized hives, and their skin tests are all negative to these foods. More commonly, because our main season is the tree pollen season, it says birch, but oak and other trees all do the same thing, and mainly cause problems with fruits that have stones in them. So when someone eats particularly a peeled fresh apple or plum or apricot or peach, they may get this itchy problem in the mouth, lips, and tongue. But if the food is cooked, like applesauce, then it doesn't cause the issue. Or many times if it's peeled, it doesn't cause the issue. And that's because these proteins that are on the foods are very sensitive and heat labile and are sensitive to enzymes in the saliva. So they get broken down very quickly or get broken down if the food is cooked or heated. Grass pollen sensitivity can be associated with this localized pollen food cross-reactivity issue with orange, melon, tomato, pea, and peanut. So just to summarize, we've talked about a variety of different outdoor pollens and mold spores that contribute to the itchy, watery eyes, sneezing, nasal congestion, wheezing that you see in your patients during the pollen seasons. Remember that the major allergens come primarily from wind-pollinated plants, not from insect-pollinated plants. So your Aunt Sally, who claims that she's allergic to lilies or to flowery, fragrant flowers, it's really not the pollen. It may be an irritant effect from the fragrance. It's really the air-pollinated plants that cause the allergy issues. Our main allergy season is a long one in Florida. Starts in December, peaks in March, and can go through into May. Primarily caused by tree pollen, mostly oak, but about a dozen other trees are all making pollen that same time of year that people can be allergic to. And particularly if people travel toward the northern part of the country in the spring, they may start with the pollen season in Florida, then hit the peak in the Carolinas in April, and continue if they go up into the Northeast to hit the peak in May for tree pollen up there. And keep in mind there's cross-reactivity among families of pollen-producing plants and also cross-reactivities between pollens and foods that cause this localized pollen food cross-reactivity issue. Most of the time, if we skin test patients to these foods, they'll be negative, so they can eat the food if they wish, if they tolerate it, or maybe more tolerable if the food is cooked or peeled. So with that, I guess I'll end, and I'll just peruse you through some of the references that have come out of the Division of Allergy and Immunology over the years. And all of these are related to pollen identification from faculty and fellows from really from the 80s and 90s, even up through recent years. So this takes, a lot of energy and effort. And remember, if you're going to be practicing allergy in an area that's new to you, you really need to take some time to learn the important allergens and seasons in the area where you're practicing. So with that, I'll end. And if anyone has questions, I'll be happy to answer them.
That was fantastic, Mark. Really, really incredible run of you went the complete gamut with this presentation. Thank you so much. And, those closing credits really, it's just a testament to what Sam Buchans and Dick Lockey have built here over what's been 51 and a half years. Just absolutely incredible. And what you yourself and Dennis Ledford, still Dick Lockey, Farnaz Tabatabian, Juan Carlos Cardet, Tom Casale, Amber Pepper, Stephanie Hootie, everybody who is involved in allergy, Song Cho, our interim division director in allergy immunology. Really, what a testament to everything here. And I saw Roger Fox is on today, one of our great faculty who retired just a few years ago from allergy immunology. I love those closing credits because it is truly a testament to the incredible scholarly activity of this group and its contributions to the world. And of course, Dr. Lockey being head of the World Society, Dennis being head of the National Society, the books that have come out, that have been written by this faculty, nothing short of spectacular. So really, I wanted to take a moment to just recognize the division for all of its spectacular contributions to science. And what a for our residents, what an incredible nature for you guys to have this opportunity to have such great faculty here, international presence faculty here. I couldn't stop thinking about that elm tree and having a blanket with that beautiful day for that picture of the elm tree. So, and that for our internal medicine residents, of course, they would be with a Harrison's book of textbook of internal medicine out there, or with my Lazama book of internal medicine learning, much easier to carry, lighter weight, and easier to reflect quicker. With that, we'll open it up to questions for this great presentation. I'm sure Dennis or Roger has a question. Nothing yet. Everybody's astounded by the completeness. I love the microscopic too. Thank you for doing that. That was great. The macroscopic and the microscopic was fantastic. I love that sequence. Jen, did I miss any comments? I think we've got the code in there. The CME code is in there.
CME code is in there, and we're good on chat.
Fantastic. And
I can forward any questions that come after.
Okay, we'll forward any questions that come to you, Mark. Thank you again. I can't believe it's been 21 years. Time flies when you're having fun. Mark and I see each other often at the VA here, since our offices are in the same floor. So always a pleasure chatting with Mark, and Dr. Cho and Dr. Ledford also are here. So I've decided to plant myself close to mast cell and eosinophil central. And now we know why I'm sneezing so much. The residents, Dr. DeGennaro now knows why his attending, Dr. Lazama, has been sneezing so much this week. So that's good. I appreciate it. All right, with that, we'll let it